Okay. So hello everyone. This is the second video of my lecture series. So in previous video we have studied about the cell, what the definition of cell, the structure of cell, both animal cell and plant cell. Okay, what are the component of cell? What is the organelles present in both plant cell and animal cell? Okay, so in this video, we'll study about the detailed structure of prokaryotic cell. Okay, this is a type of cell which are unicellular in organism. Both prokaryotic and eukaryotic organisms are the types of cell. That is, prokaryotic cell is the primitive, and eukaryotic is the advanced cell. Okay, in primitive there is no nucleus, there is no organelles. We have studied in the previous video. Okay, so domain, we know about the organized three domain system, right? What are these? Let's see. These are three types of domain. Okay, first, domain eukarya. Second, domain archaea. And third, domain bacteria. Okay, domain bacteria. So domain archaea and bacteria are the prokaryotic organisms only. Prokaryotic organisms is under the domain of archaea and bacteria. Domain eukaryotic organisms. Okay, these are the three domain system in the classification. Okay, three domain in the classification system. Okay, then definition: prokaryotes are single cell organisms. We know prokaryotic cells are the unicellular organism, that is single cell organism, that is the simplest organism, that are the most primitive forms of life on earth. Okay, so most of the prokaryotes are extremophiles. Okay, what are the extremophiles? So, an extremophile is an organism that is able to live in extreme environments. Okay, that is environment which condition like extreme pH, extreme salinity, extreme temperature. These are the extreme conditions for the growing of bacteria or growing of prokaryotic organisms. Okay, we live in the natural or general condition, that is no extreme temperature, no extreme pH. Okay, but the prokaryotic organisms are live in the extreme levels, extreme conditions only. Okay, and that they are found almost everywhere. They are the ubiquitous organism. Okay, they are found in everywhere, in our mouth, in our skin, in our palm, in our finger. Okay, it's there almost everywhere, they are found. Okay, and fossils show that prokaryotes are already here on Earth 3.5 billion years ago. Okay, so as we know, the primitive organisms are the prokaryotic organisms. So they were already here on the Earth. Okay, from the prokaryotic organisms, lives rise to all of the life form present on Earth today. Okay, the scientists think that prokaryotic ancestors gave rise to all of the life forms present on Earth today. Okay, then next, this is the structure of prokaryotic cell. Okay, see, this is the pili. Okay, this is the pili. You can see the all over the cell, all over the what, all over the outer membrane of the cell or a cell wall. These are the pili. Okay, and this is the ribosomes, and this is food granules. This is the prokaryotic flagellum. Okay, we'll study the function of pili, the function of flagellum, what is ribosomes, what is food granules. Okay, so This is the plasmid. What is plasmid? It's a plasmid is the extra chromosomal DNA. Okay, extra chromosomal DNA is called as plasmid, and this is also the double-stranded DNA. Okay, can replicate independently. Okay, it can replicate itself, and this is the cytosol. This is the plasma membrane, cell wall, and the outermost is the capsule or slime layer. We will study about the capsule or slime layer, cell wall, plasma membrane. We will study all these terms in the video lecture series video. Okay, then the cell wall. Okay, cell wall is the outermost covering layer of a cell. Okay, see, cytoplasm of prokaryotic cell has a high concentration of dissolved solute. Okay, hence the osmotic pressure is high. Okay, the prokaryotic cell is high concentrated concentration of dissolved solute. Okay, so because dissolved solutes are high concentration in the cell, hence the osmotic pressure also high. The cell wall is a protective. That's why the cell wall is a protective layer. Okay, that surrounds some cells and gives them shape and rigidity. Okay, because of the high concentration of dissolved solute, they can burst at any time, right? So hence because of osmotic pressure is high also, so it can burst. That's why the to give the protection to the cell wall is present, and also it gives the shape and rigidity. Okay, bacterial cell walls contain peptidoglycan composed of polysaccharide chain. Okay, bacterial cells contain peptidoglycan composed of polysaccharide cells. S layer that is surface layer, that is also paracrystalline in structure. Okay, proteins are also present on the outside of the cell walls of both archaea and bacteria. Okay, protein. What protein? That is the S layer protein. That is the surface layer proteins are also present on the outside of the cell wall of both archaea and bacteria. Basically, archaea and bacteria means what? Means prokaryotic organisms. So S layer proteins present on the prokaryotic organisms. Got it? Then bacteria are divided into two major groups, that is gram positive and gram negative bacteria. Here see. Okay. First, mm, two, that is gram positive bacteria and gram negative bacteria. Okay. How can we distinguish both bacteria, that which is gram positive and which is gram negative? Okay, we will apply the staining technique. Okay, by the application of the staining technique or gram staining technique, we can distinguish the both, which is the gram positive bacteria and which is the gram negative bacteria. Okay, see. Okay, here is the gram staining. What is gram staining? How can we distinguish the gram positive and gram negative bacteria? By the application of gram staining. Okay, in a plate we took the both. We don't know na, we even don't know what is gram positive, which is the gram negative bacteria. So we took all the bacteria from the culture plate to the to a culture to a normal plate for staining. Okay, then this is the microscopic view. See, this is the microscopic view of both gram positive and gram negative bacteria. This is a mixture of both gram positive and gram negative bacteria. Okay, this is the microscopic view. And then we applied a crystal violet. Why? Why we applied the crystal violet? Because crystal violet is a positively charged, and therefore it can adhere to the cell membrane. Okay, because of the cell membrane is in outer is the negatively charged, and crystal violet is the positively charged dye. Okay, that can easily adhere to the cell membrane. Okay, that's why we took crystal violet. Okay, then after 60 seconds we took iodine orange. Why iodine orange? Why not safranin? Why not sodium chloride? Why not potassium chloride? Why we took iodine? Because iodine is used as the mordant that causes the crystal violet to penetrate to the gram positive bacteria. Okay, it helps in the penetration of the what crystal violet. Okay, first we applied crystal violet na, so iodine can penetrate the crystal violet to the gram positive bacteria. Okay, this is the gram positive bacteria. Then remains are the gram negative bacteria. Okay, then okay ठीक. Then we applied after 60 seconds we applied ethyl alcohol for 5 to 10 seconds. Okay, why why we applied ethyl alcohol? Because ethyl alcohol is a transparent that can decolorize the both the bacteria, decolorized. Okay, with both the bacteria, no, it can't decolorize the gram positive bacteria. Why? Because almost the penetration of the crystal violet was occurred by the help of iodine. So how can ethyl alcohol decolorize it? Okay. Then ethyl alcohol for decolorization. Okay. Then after safranin is applied. With the help of safranin, gram negative bacteria can be easily distinguished. Why? How can it easily distinguish by the application of safranin? Because it directly stain the decolorized bacteria. Okay. It directly which bacteria are decolorized by the application of ethyl alcohol, it can stain the decolorized bacteria. Got it? Okay. Then see. This is the cell wall of gram positive and gram negative. Okay. This is the gram positive and this is the gram negative. Okay. Okay, then in gram positive bacteria the cell wall is made up of what? Peptidoglycan. And the cell wall is thick in comparison to gram negative bacteria. The cell wall of gram positive bacteria is thick. Why? Because the presence of peptidoglycan. Okay, peptidoglycan present in the gram positive bacteria's cell wall and in a thick layer manner. Okay, and also in gram negative bacteria there is also peptidoglycan is present but in a thin layer. Okay, then outer to the peptidoglycan in gram negative bacteria, that is presence of lipoprotein. Can we see the lipoprotein in gram positive bacteria? No, no lipoprotein in gram positive bacteria. Okay, so outer to the lipoprotein there is also present the lipopolysaccharide. Oh, lipopolysaccharide. Okay, got it? Then this is the plasma membrane. Okay, this is the plasma membrane in both gram positive and gram negative. Okay, then this is the cell. Okay, and this is the cell of gram positive bacteria, and this is the cell of gram negative bacteria. Oh, okay. That's why we can distinguish the gram positive and gram negative bacteria because of the composition, the different composition of the cell wall. Okay, we can distinguish the bacteria gram positive and gram negative by the help of staining technique. Okay, staining, gram staining, we first we applied what? First we applied crystal violet, then we applied iodine. Okay, then we applied what? Ethyl alcohol, 95% ethyl alcohol. We can prepare 95% ethyl in laboratory also, because this the staining technique is occurred in the lab na. So we have to prepare the ethyl alcohol, 95% ethyl alcohol. Okay, 95. Okay, also 95% ethyl alcohol also manufactured. Okay, they can available in market also, 95% ethanol. Okay, otherwise you will what, you will prepare yourself 95%, 85%, 70%, 60%, whatever percent it may be, you will you can prepare yourself. Okay, okay, 95% ethanol we took. Then what? Then saffronin. Okay, acha. Then we can distinguish the gram positive and gram negative bacteria. Okay, we distinguish the gram positive and gram negative bacteria. Then we see what? Then we saw that the cell wall composition of gram positive bacteria is the peptidoglycan. There is a thick layer of peptidoglycan. Okay, and in gram negative bacteria, also peptidoglycan is present, but in a thin layer. Yes, also in gram positive bacteria, 90% of gram positive bacteria's cell wall is composed of peptidoglycan, but another 10% is composed of what? Teichoic acid. What? Teichoic teichoic acid. But in gram negative bacteria, no teichoic acid. No, there is no teichoic acid. Only peptidoglycan in a thin layer. Outer to the peptidoglycan lipoprotein and lipo. Then outer to the lipoprotein, that is lipopolysaccharide. Okay, got it. And then okay, yes. This is a different bacterial responses to the staining procedure are ultimately due to the cell wall structure. Okay, got it. Gram positive organisms typically lack the outer membrane found in gram negative organisms. Okay, clear? Then see here. This is the gram positive bacteria, right? This is the gram negative bacteria. Okay. Then this is the plasma membrane. Okay, plasma membrane also present in gram negative bacteria. I have mentioned. Then this is the peptidoglycan, which is present in a thick layer manner in gram positive bacteria. Okay, but in gram negative bacteria, that is only one. See, one, two, three, four, five layers of peptidoglycan. But in gram negative bacteria, okay, one layer of peptidoglycan. See, got it? Then the outer membrane is what? From the peptidoglycan also the outer membrane is present, and the outer membrane is the lipoprotein. Acha, lipoprotein. Then to the outer of lipoprotein there is a lipopolysaccharide also present. Okay, I have mentioned here na. See, I have mentioned here. This is the gram negative lipoprotein and lipopolysaccharide. But in gram positive there is no lipoprotein, there is no lipopolysaccharide. Okay, see. Okay, clear, understood. Then next, okay, the plasma membrane. Acha, to the plasma membrane is the innermost layer of the cell wall, right? Because the cell outer membrane is cell wall, na? To cell wall to inner, this is the plasma membrane. First, see, okay, plasma membrane. C double one O. Okay, O C H two. Okay, then C double one O O C H two C O P double one O O. That is the word. Okay, then see why I draw here. See, you will understand. Okay, because of this. Okay, plasma membrane. The plasma membrane is a thin lipid bilayer. Okay, thin lipid bilayer, 6 to 8 nanometers, not more than that. Okay, that completely surrounds the cell. Okay, that completely surround, not halfly or partially. Okay, completely surround the cell. Its selective permeable nature keeps ions, proteins, and other molecules within the cell. Okay, that is a selective permeable membrane. Okay, we'll discuss what is selective permeable, what is semi permeable, and what is permeable. Okay, then now see phospholipid from archaea. Okay, this is the phos- why phospholipid? Because a phosphate group is present in the lipid. Yes or no? This is the phospholipid from archaea, and this is phospholipid from bacteria and eukarya. Okay, then the plasma membrane of bacteria and eukarya structure is same, like phospholipid bilayer from bacteria and eukarya is same, and the phospholipid bilayer from archaea is a little bit different. How different? You can see this is the what? This is the what? Glycerol, right? CH2C, like CH2. Okay, then C, then CH2. Okay, here glycerol present. Okay, in phospholipid from archaea glycerol is present. Okay, in phospholipid from bacteria and eukarya, glycerol, each glycerol present. Yes, glycerol is present. Okay, is phosphate present in phospholipid from archaea? Yes, present phospholipid. That's why we call phospholipid, na, because of the phosphate group, presence of phosphate group. So, is the phosphate group present in phospholipid from bacteria and eukarya? Yes, present. But is it ester linkage is present in the archaea? No, ester linkage is present in what? In bacteria. Okay, this is the difference between the phospholipid from archaea and what? Bacteria. There is the ether linkage in phospholipid, and that is the ester linkage in what? Phospholipid from bacteria. Got it? That's why I draw this diagram. Then see what is this? Okay, this is the glycerol, right or wrong? This is the phosphate. Okay, glycerol, and this is the phosphate group. Okay, this is what? This is phosphate group, and this is the ester linkage. Okay, what is this? This is the ester linkage. Ester linkage. ESTER. Ester linkage. That is the plasma membrane structure of what? Bacteria. Bacteria and eukarya. Then in sorry. Okay. All are same phosphate group. Yes, this is phosphate group is present. Glycerol, yes, glycerol is present also. But in the place of ester, this is ether group is present. What? Ether group is present in archaea. Okay, and then this is bacteria. Got it? Okay, then see. Okay, this is a phospholipid bilayer from bacteria. In bacteria, that is ester linkage is present, and in archaea, that is ether linkage is present. Okay, got it? Okay, then summary. What we have studied in this lecture. First, we have studied the prokaryotic cell. That is prokaryotic. So these are the what? These are the simplest organism, and these are the unicellular organisms, single celled organisms, and these are also called the extremophiles. Okay, okay. Then this single celled organism. Then these are called the extremophiles. Okay, got it? Single celled organisms, and these are called the extremophiles. Okay, then we have studied about what? The cell wall structure. Okay, the typical prokaryotic cell structure we have studied. Then flagella, pili, ribosomes are also present. Then we have studied about the cell wall, and we have studied about the what? Gram staining technique, how we can distinguish the gram positive and gram negative bacteria by the application of gram staining technique in laboratory. Okay, okay. Then we have studied about the cell wall, right? What are the difference between the composition of cell wall in gram positive and gram negative? Okay, then we have studied the plasma membrane. right? Studied about the plasma membrane, the structure, and in which domain the ester linkage is present, and in bacteria and eukarya, and in archaea domain, that is the ether linkage is present. But in both the what in both the group, the phosphate and what glycerol is present. Okay, so we'll see in our next video. Okay, thank you, bye.